CRISPR-based gene editing of telomere-related genes represents one of the most advanced and potentially transformative frontiers in the science of aging. At the heart of this emerging technology is the capacity to directly modify the genome with unprecedented precision, allowing researchers to activate, silence, or repair genes that control cellular lifespan and telomere integrity. The most promising target within this field is the HTERT gene, human telomerase reverse transcriptase, which encodes the catalytic subunit of telomerase, the enzyme responsible for maintaining telomere length. In theory, CRISPR could be used to safely upregulate HTERT in somatic cells, reactivating the cell's own telomere-preserving machinery. Telomeres are repetitive DNA sequences that cap the ends of chromosomes, protecting them from degradation and preventing unwanted fusion with other chromosomes. Each time a cell divides, these telomeres shorten, eventually leading to cellular senescence or apoptosis when they become critically short. In stem cells and certain immune cells, the enzyme telomerase prevents this attrition, allowing for continued replication. However, in most adult somatic cells, HTRT is silenced, meaning telomerase is inactive and telomere shortening proceeds unchecked. This process is a key driver of aging and age-related disease. CRISPR, clustered regularly interspaced short palindromic repeats, especially in its CRISPR-Cas9 form, enables scientists to make highly specific edits to DNA. In the context of telomere biology, this opens the door to upregulating HTRT expression or correcting mutations in genes like TRC, POT1, and TRF1/2, all of which are involved in telomere maintenance and structure. Animal models have already demonstrated that CRISPR-based reactivation of telomerase can extend lifespan, regenerate tissue, and improve organ function without immediate side effects. These findings mark an exciting shift toward the possibility of treating aging not just as a passive decline, but as a programmable and modifiable condition. One particularly promising approach involves CRISPR, CRISPR activation, a variant of the traditional CRISPR system that doesn't cut DNA, but instead uses modified Cas proteins to upregulate specific genes. By using CRISPR to enhance HTRT expression in a controlled and tissue-specific way, researchers may avoid the risks of full-blown telomerase reactivation, which in some contexts could increase cancer risk. For example, in immune cells or endothelial cells, both of which suffer functional decline due to telomere erosion, temporary or regulated HTRT expression could restore vitality and resilience without uncontrolled proliferation. CRISPR can also be used to study the epigenetic regulation of telomere-associated genes. By targeting histone modifiers or DNA methylation sites around the HTRT promoter, scientists can investigate how aging and environmental stress turn these genes on or off. This insight may lead to therapies that restore telomerase activity naturally, without permanent genomic changes. In fact, combining CRISPR with epigenetic reprogramming technologies like Yamanaka factors is now being explored as a way to rejuvenate cells while preserving identity and function, essentially rolling back the clock without triggering cancerous transformation. Though the promise is extraordinary, the technology is still in its early stages and significant hurdles remain. Chief among them are safety concerns, especially regarding off-target edits that could disrupt other important genes or activate oncogenes. Additionally, the long-term effects of telomerase reactivation are not fully understood. While telomerase can extend cell lifespan, it can also facilitate the survival of damaged or precancerous cells if not carefully regulated. Thus, any CRISPR-based therapy targeting telomere genes will require extremely precise control over when, where, and how long these genes are activated. Ethical considerations are also front and center. Unlike nutritional or pharmacological approaches, gene editing carries implications for germline transmission if applied to embryos or reproductive cells. Although current telomere editing research focuses strictly on somatic cells, the boundary between therapeutic use and enhancement remains a topic of debate. Nevertheless, as aging itself becomes increasingly viewed as a modifiable biological process, public attitudes towards such interventions may shift, particularly if they offer the potential to reduce suffering and extend health span. Despite these complexities, CRISPR-based telomere editing continues to move forward. Several research groups are developing delivery platforms such as lipid nanoparticles and viral vectors that can carry CRISPR components safely into specific tissues. Others are working on inducible systems that only activate gene editing in the presence of certain drugs or conditions, minimizing unintended effects. Together, these innovations are helping build a future in which telomere biology can be precisely and safely manipulated to slow or even reverse aging at its root. In summary, CRISPR-based editing of telomere-related genes is one of the most exciting and ambitious areas in longevity science. While still experimental, it holds the potential to directly restore telomerase activity, stabilize chromosome ends, and rejuvenate aging tissues. With further refinement and oversight, this technology could eventually offer a revolutionary approach to aging, less as a fate and more as a condition we can learn to manage, repair, and even reverse.